Okay, Miko, do you think we can start? We're already a few minutes past 10.
Yes, we can start, yes.
Right, so good morning, everybody. Welcome to the CFTC weekly seminar. It's a pleasure to host Pablo Sartori from Instituto Gulbenkian, Ciência in Lisbon. Pablo is originally from Spain, where he studied at the University of Granada. He then spent some time in the US, first at IBM Research and then Rockefeller University. Oh, sorry, I'm getting this in the wrong order, aren't I? He did his PhD at the University of Dresden in Germany, and then he spent some postdoctoral time first at IBM in New York and then Rockefeller University. And after that, he came back to Europe and he joined Instituto Gulbenkian de Ciência, where he is doing the research that he will tell us about today. So, Pablo.
Yeah, so is the screen sharing still going on?
Warmly welcome, and you can start anytime you're ready.
Can you see this?
Yes, we can see it, but yeah, if you could go into full, yeah, we can see it, all right.
It's in full screen mode now.
All right, okay, very good.
Yes, well, thank you everybody for those of you who are here for attending this seminar. Please feel free to interrupt me whenever you want. I think I'm aiming for sort of like 30-35 minutes of uninterrupted speaking. So if anything, we will finish early. Yeah, first of all, I'd like to apologize for having had to postpone the date. That something unexpected happened. I mean, other than my daughter, now I have a little son, so he came a bit ahead of time, and that's why we have to cancel. So I think I have for once, a very good excuse for such things. As it was just mentioned, I am currently working at the Instituto Gulbenkian. So for those, I don't know how well known this is among the physics community in Portugal, but this is a biology institute. But I'm a theoretical physicist by training, and until last month, I was, although not physically, I was still working at Rockefeller University and in the Institute for Advanced Study. Okay, so I had some kind of joint postdoctoral position. I oscillated between the two. So it's been quite a, Rockefeller University is a biology institution. IAS is a predominantly, I would say, a theoretical physics and mathematics institution. So I thought maybe a good place to start is with a little anecdote. People like this type of anecdotes, and I enjoyed this one of something that happened relatively recently at the Institute. This was less than 1 year ago. It was, so you see, at IAS organized regular dinners, and last fall I happened by luck to be sitting next to Freeman Dyson, who was to my right. And to my left there was some other physicist whose name we don't have to remember now. And you know, even though I know Freeman a little bit because I just exchanged a few words a couple of times because I know Joel Lebovitz a bit more and they're friends, I was very shy to ask anything, but I was lucky enough that this physicist did ask something related to my own work, and he said he was wondering about Freeman's opinions on biophysics, right, and all the progress that seemingly had happened in recent decades on the biophysics, theoretical biology done by physicists. Freeman gave a perhaps surprising answer. He says the progress in biology is truly amazing, but of course some people are spoiling it by trying to make biology look like physics. Okay, and needless to say that the other guy was a bit shocked because he had a theoretical physicist, a very talented mathematician, a very renowned person that somehow is dismissing physicists' attempts to tackle biology. And I like this anecdote because I think it's good to remind ourselves, or myself as a physicist working in biology, that you have to be very critical about the progress you're truly making. Okay, it's very hard to assess progress in biology. It's very hard to not make it into some very, very applied physics to very concrete problems. Okay, so. And at the same time, if you try to make very general things, it's often the case that you just end up talking about nothing, right? Philosophizing. So it's very hard to find a middle ground. So when I pose questions like this, which are questions I'm interested in, what are the phases of cellular matter, right? What are the phases that describe the state of matter of a cell? You know, I tend to be a bit skeptical of physics, classical physics answers, like, you know, has to do with a liquid or with an elastic material or with a liquid crystal or with a glass or something like that, right? And I tend to like biologists' answers, which says things like a cell is a bag of proteins. Okay, so it's obviously very heuristic, but I like this answer. Although even though it's heuristic and not very precise, I think it misses one important point, which is within the cell you have proteins diffusing around, you know, as in a bag of proteins. But this proteins, right, which are these structures, these large molecules, they coexist with higher order structures, right, with aggregates, which are called protein complexes. And these protein complexes are stable, and at the same time they coexist with these diffusing proteins, and they have the, you know, they exhibit the typical problem that you face when you try to work in biology as a physicist, which is very diverse, right? So they're diverse in function, in size, in composition, in lifetime, in almost every property, right? And there goes the classical problem: how can you develop theory beyond modeling each individual system, right? So a temptation is to do a very good model of one particular protein complex, but I think we ought to do something a bit better than that, although it's not clear how far we'll manage. Now, an analogy that I like to bring up in this context is the history of thermodynamics. Right? It's one of the beautiful pieces of science history and physics, and it has to do with how out of these steam engines that were developed at the late 18th century, we developed a very powerful, very formal today science, right? So although people knew how to design very sophisticated engines, they were messy, they were loud, and they had many different parts, right? They had governors, they had pistons, they had phase transitions going on, they had cogs. And it took the bright mind of Sadi Carnot to replace all these complicated diagrams of heat engines by something very, very simple, which is essentially, after 200 years, the same diagram that we use today for heat engines. And once he could give this leap of abstraction and essentially choose the right variables, right, to describe a heat engine, he could easily develop, he could develop what today we call the Carnot cycle. And soon after, I mean, his work was a bit lost, but pretty soon after it was recovered and the thermodynamics was formalized, right? And it's quite astonishing, no? That from a theory, I mean, it wasn't clear there was even a theory to be done, right, of steam engines, right? It's this very noisy, dirty machines that are used by engineers, and then something elegant that has been very, very robust for 200 years emerged, right? So, I mean, that's kind of a dream for biology, but it's an inspiring dream, right? And I think that the key is that when you have a complicated structure like a protein complex, you need to know what are the variables that you ought to choose to describe it, right? And then maybe if you choose the right variables, you can begin to distinguish the different phases of these systems and understand the boundaries between these phases and even hope for some degree of universality in your conclusions, right? I mean, probably won't be universality in the RG sense, but some kind of, you know, maybe, you know, something maybe like an ideal gas law, something like this, right? I mean, it's not clear that even that will be true, but we can hope. At least we ought to be able to choose the variables, right? Okay, so since I think not everybody in the audience is an expert in biology, and probably neither am I, I'll just say a couple of words of what a protein is, at least for the purposes of this talk, which I hope are at least illustrative. So a protein is a polymer, okay? It's that has about 360 amino acids. I mean, well, about 300 amino acids, typical protein of a bacterium. This is a lactopressor protein, which has 360. It's a famous protein. It doesn't matter why today. And it contains 20 different amino acids, okay? So each amino acid is depicted in a different color. Each amino acid has about 10 atoms. And it's interesting that these are very typical numbers for proteins. Okay, so as I mentioned, 300 in length is a typical size of a bacterial protein. And 20 amino acids in composition is a typical composition of a protein. And what this means is that if you define a variable called heterogeneity, which is the ratio of the different number of components, which in this case is 20, over the total number of components, which in this case is on the order of 300, you get a relatively low value. Okay, so proteins have low compositional heterogeneity. So that's something to remember. Another thing to remember is that these 20 amino acids that you find in most proteins are not too mysterious. I mean, there are only actually 20 amino acids in the cell. Okay, so that's why they use 20. But it's not mysterious, but it's also not trivial. So you could imagine a different world, or a different set of cells in which, out of this alphabet of 20 amino acids, some proteins use 5, others use 3, others use all 20, or they all use 10, each one 10 different ones, right? But by and large, this is not what you see. What you see is that all proteins use the whole alphabet at their disposal. Okay, so they make a dense usage of the alphabet. And with this dense usage and this low heterogeneity, the cells manage to do one of these beautiful tricks of biology, which is combinatorial expansion. And out of modest 20 amino acids alphabet, you have thousands of proteins. Okay, so within a cell, you find typically thousands of different protein species within E. coli, for example. Okay, so low heterogeneity, dense alphabet usage, and strong combinatorial expansion are the properties of proteins as assemblies of amino acids. So what about protein complexes? It's what I want to talk about today. So protein complexes, I like to think of them as just a change of units, okay, a change in scale. So a protein complex is an assembly of multiple proteins. I mean, you can see now it's much more fuzzy, the atomic structure, right? Because we're at a much larger scale. And this is a classic protein complex as well as a polymerase complex, not too important why it's so famous. This is from bacteria, and it's relatively small. It has 6 proteins. And among these 6 proteins, there's 5 different ones, okay? So there's only, there's 5 different colors in this picture. It's hard to give a good estimates for the sizes of protein complexes, but tens of proteins is a good order of magnitude, I think, right? And what's perhaps more interesting is that the composition is often comparable to the system size. So that means that if we define again the same quantity, the same variable, the heterogeneity, as a number of different components over the total number of components, we get a number of order 1. Okay, a number of order 1. And that's different from what we observe for proteins as assemblies of amino acids. Now, the alphabet for protein complexes is very large. It's the whole proteome. Okay, so within a cell, you have thousands of proteins, and each protein complex could have used any of these thousands of proteins to assemble. Yet, what we find is that protein complexes are relatively small compared to the alphabet, right? So the usage that they make of the alphabet is very sparse. So each complex uses a small fraction of the proteome. Okay, it need not be that way. You could have a different world in which the usage was dense as for proteins, but that's not what we observe. And finally, another surprise is that there's no universal or no generic combinatorial expansion of the protein, okay, in this sense. So you don't find that from 1,000 proteins, you obtain a million or 10 million protein complexes. You find that you obtain about 1,000 protein complexes. So somehow this beautiful trick of biology, which is playing combinatorics, it seems to be missing here. Let me remove this so I look a bit less weird. Okay, so what's going on, right? There's no combinatorial expansion. Let me say 2 more words about that. What I mean is that you could imagine, if I were to naively design the world of cellular self-assemblies, I would have chosen that have 5 proteins. I can assemble them in a certain way, such that they perform a certain task, RNA synthesis, for example. And then I could assemble them in a different way, so that they perform a different task, such as RNA degradation. Okay, this seems like a good idea, but it's not what you see. Okay, you don't see this. another more playful way of thinking of it is that cells don't play protein tangrams, right? So you probably have, many of you have probably played this game or at least know of it, but you have seven pieces and you play to assemble them in different shapes. And this seems to work fine when you do it on your desk, right? But for some reason at the cellular scale, this doesn't happen, and there must be a reason why this doesn't happen, right? So it seems like such a powerful strategy. So I guess what I'm trying to say is that I have a proposal for what can be good variables to understand protein complexes, which can be things like the heterogeneity, the usage, right? Something that we haven't used yet, but something like an expansion. And we may have an idea of where in the phase space of these variables protein complexes lay, right? So they seem to have high heterogeneity, sparse usage, weak combinatorial expansion, and it's not a priori obvious why this is the case, given that proteins, they exist in a completely different region of this phase space, if you wish, right? So at a different scale, this doesn't seem to work. And now once we have these variables, we can start asking questions such as fundamental biological questions of things, such as how many complexes can a proteome assemble, right? What is the limit for a cell? What determines the heterogeneity of complexes? I mean, why are these more or less heterogeneous? And why don't they play tangrams? Are there physical constraints for that, or is there another reason? Okay, so the way we try to address these questions is developing a statistical mechanics model. It's a bit of an odd statistical mechanics model because it's suited for this particular problem, which I think makes it also very interesting. Okay, so I think this even if you don't care at all about biology or protein complexes, I think there's substantial new statistical physics to be learned in this direction. Okay, so in our model, what we have are components, which are this. By the way, I emphasize again, please feel free to interrupt me whenever you want, and especially in this slide. So we have what we're calling the proteome, right, which is the ensemble of all different protein species, labeled by P from 1 to N prot, right? So it's a certain size, and there are these little gray squares. And what we want is that this proteome assembles into complexes. And in our model, a complex 1, complex 2, complex capital K, is a square arrangement of proteins, okay? So it's a square made of numbers, okay? Now, this square made of numbers, these complexes have a certain size, and they have a certain composition, and they're characterized by who is a neighbor of whom, right? And although this is a very simple setup, it already includes all variables that we introduce, right? So the types of complexes, capital K, the proteome size, the size of a complex, the composition of a complex, and therefore the heterogeneity and the usage. Now the question is, how can we develop a thermodynamically consistent model that would allow the proteome to assemble into a certain number of predefined complexes, okay? And a given proteome shall always assemble into the given complexes and no others. So one way to do this is borrowing an idea from the neural network literature. I mean, I say this because it may resonate with some people. So what I'm going to talk about is, so this type of way of defining the binding affinity is related to models of neural networks developed in the 80s by John Hopfield and later by others. And the idea is that we establish the binding affinity of two protein species, P and P prime, based on the complexes of which they are to form part. So the binding affinity of protein P and P prime is -E. E is an energy scale. If P and P prime are neighbors in a given complex, so for example, 23 and 1 are neighbors in complex 1, and so their binding affinity is -E, but 23 and 20, they're not neighbors in any complex, and so the binding affinity is 0, okay? And so now we can have a proteome, we can define complexes, we can define a binding affinity matrix, and we can tune the statistical properties of complexes, right? So for example, we can have complexes have the same protein several times, right? Here I underlined 23, so you see 23 appears several times in the same complex, twice in this case. And it can also be shared among different complexes, right? And this will modulate heterogeneity usage and so on. So we can control the statistical ensemble and play with this rule for binding affinity and see what we get. Okay, so we then did a 2D lattice implementation of this model, a numerical, and we simulated in the, so is this, I know nobody's gonna say yes, but I hope this is more or less clear. If it's not, please feel free to interrupt. So we encoded this in the, we programmed this with the grand canonical Monte Carlo simulations, and I'm just gonna show you some of the results, right? So we start by a very simple case in which our proteome encodes one single complex. And then we try to do a nucleation experiment. So we put a small fragment of the complex we want to nucleate. And then we run the simulation and see what happens. And what happened is that nucleation occurred, right? So the fragment is larger than the critical nucleation seed that you can estimate. And the full complex assembled, right? So this full complex, it's depicted here as a red square, but that's just, which is a bit unfortunate, but I couldn't come up with something better. But you have to understand that it's really very heterogeneous in composition. Okay, so each pixel here is really a different protein species. I didn't assign colors to protein species because you wouldn't see anything, right, if I did so. Okay, so it seems that when we encode one complex in our proteome, things work out and self-assembly happens. So then we went to the natural next step of encoding with the same proteome, more complexes, in this example, 8. And what happened is that self-assembly didn't occur, right? So a similar fragment, we let the simulation run. And what happened is that the complex started assembling, the red complex started assembling, but then a chunk of the blue complex attached, and then a chunk of the yellow complex attached, and then you have this unbounded growth that somehow percolated over the whole simulation space, and it got stuck there, right? It got the simulation sort of didn't evolve very slowly thereafter. So what's going on? I mean, somehow self-assembly seems to work for one complex, self-assembly seems not to work for many complexes, and you have this chimera structure that we call it chimera because it's chunks of different, finite chunks of different complexes. So what happens is that our naive model has a rather rich phase space. Okay, I'm gonna use the word phases, although these are not really thermodynamic phases, okay? In the sense that we're not obtaining the thermodynamic limit. There should be a tessellation of the space in different self-assemblies, but yeah, I realized right now, I forgot to change the name to regimes, which is what I normally write when I talk to physicists. I use phases for biologists because they relate more to that word. Anyway, so there's a self-assembly regime, right? There's a chimera regime, and there's also a liquid regime, which is a dense phase in which components constantly rearrange, and there's a dilute solution regime, sort of like a vapor phase. And we can access the different phases by changing thermodynamic parameters, such as binding energy and chemical potential, and using the correct ordered parameters, right? So it's a bit phenomenological, as we call it the assembly error, is just what fraction of the assembly was retrieved. So it's not too important. And then we can even have analytical estimates for the slopes of the phase boundaries, right? When you talk about chemical potential and binding energy. So more or less we could understand this phase diagram when we use thermodynamic variables. But what we were interested in was understanding this transition between self-assembly and chimera, because what I showed you before was that you can elicit this transition not by changing temperature or binding energy or chemical potential, but by instead changing statistical properties. Such as how many complexes your proteome encodes. So a bit the idea is the following, right? What happens if you try to encode too many complexes, you will have a transition to chimera. And the argument goes as follows. If you have a finite proteome and you try to assemble more and more, one complex with it, things may work out. But if you try to assemble more and more complexes, you will be reusing the same proteins in many complexes. And that means each protein will have many, many different binding partners. Okay, and this is called promiscuity in biology, right? So how many different binding partners a certain protein has. And so if you increase the number of binding partners, eventually, as self-assembly is happening, a protein may bind the wrong partner, and a chimera may happen. And what this means is that the number of complexes, how many types of complexes you assemble, capital K, it will be limited by a certain sheet, right, a certain manifold, K max, which will depend on the remaining statistical properties of our problem, right? So the heterogeneity of complexes, the usage to make of the proteome, and the size of the protein. Okay, so let me just give you a bit of a more precise argument for one particular case. So consider that you have N-prop proteins, okay, or components that are playing tangram with K complexes. So you have N-prop proteins that they need to assemble into capital K complexes. Let's estimate together the probability that an error is made during assembly. So in principle, you have the red complex self-assembling, and it's growing layer by layer, right, like a normal nucleation process, and you have these two proteins, 21 and 14, in a corner of the complex, and there's the wrong protein, 42, that approaches. In principle, it doesn't belong to that complex, but it may bind there by mistake. So what is the probability that such an error occurs? Is the probability that it binds by mistake 21 times the probability that it binds by mistake 14 times the number of proteins, that's like 42 that there are, right, times the proteome size. So the probability that it binds by mistake 21 equals the promiscuity of 21 over the proteome size, right? So how many different partners 21 has, because it belongs to different complexes, okay? So in other words, the promiscuity scales with the types of complexes. And so if you force the error to be smaller than 1, what you obtain from this is that the that the types of complexes is limited by a maximum value, which scales with the square root of the proteome size, okay? And we can verify this scaling with numerical simulations, right? So we can change how many complexes our proteome has, and the transition point, right, will change with the proteome size, and it will change according to the scaling. So upon rescaling, the transition points collapse. Okay, so it seems that indeed K smaller than K max is an important boundary, and it explains the limitations of proteomes. The question now is, what can we learn about protein complexes from this? Right? So it seems that K has to be smaller than K max, and then the question is what determines K max. So I'll tell you, I give you one example, and now I'll give you, let's say, the full answer little by little. So one property that influences K max is the heterogeneity of complexes. Okay, so it turns out that K max scales with a positive power of the heterogeneity of complexes, and so more complexes can be encoded by a proteome if these complexes are heterogeneous. Okay, and you can see this in the phase diagram below, in which the variables now are very different from those of a normal thermodynamic phase diagram. So now we don't have temperature and chemical potential; those are frozen. Now we have things that are more interesting from the point of view of biology, such as how many complexes, capital K, and how heterogeneous they are, right, H. So as you can see, as you change K, the types of complexes, there's always a transition from the self-assembly regime to the chimera regime. So this always happens. But when this happens depends on the heterogeneity. Okay, so when complexes are very heterogeneous, the transition occurs later. So what that means is that you can encode more complexes with the same proteome if these complexes are very heterogeneous. And there's also later on a transition into the liquid phase, but I'm not going to talk about that. So in any case, if this argument is correct, what we would expect is that there is an overabundance of high heterogeneity complexes in the cell, right? And this is something that we can check. We can take this new variable that we've defined, heterogeneity, and look for databases of protein complexes, and try to see if indeed there is an overabundance of high heterogeneity protein complexes. And that's indeed what we observe, right? So it turns out it's not so easy to find a good database with compositional information of protein complexes, but with a bit of time, you can find it. And indeed, you find a very large peak of high heterogeneity complexes. There's also deviations from this peak. Okay, you can see this smaller peak around intermediate values of the heterogeneity. And there's an explanation for that, which we sought after. So what happens is that our model is, of course, very, very simplistic, and it doesn't take into account geometrical symmetries of protein complexes that are very important for self-assembly. So in reality, what we're capturing is the behavior of asymmetric protein complexes, right? Such as polymerases, but we're not capturing the behavior of symmetric protein complexes, such as this tetramer, right? And you can actually partition these two peaks and see that the peak that we explained has to do with asymmetric protein complexes, the other one with symmetric ones. Anyway, so what I guess what I'm trying to get after is that our model does capture the dominant effect, and deviations from this dominant effect can also be explained by further data analysis, right? And I'm being a bit brief on these things. You can find the details in our manuscript. So, okay, heterogeneity increases the capacity of our proteome, but what do we do once all complexes are heterogeneous, right? How can you push the capacity further? And the simplest way is to just make the usage of the proteome more and more sparse. Okay, so the Kmax, the maximum number of complexes that our proteome can assemble, scales negatively with the usage. So the smaller the usage of the proteome, the more complexes you can assemble, right? There's a divergence. Of course, this divergence is regularized by the finite size of the simulations. I mean, there's just scaling arguments. And you can see this in this space diagram, right? So as the usage goes down, the number of coexisting complexes that can occur in the self-assembly phase grows. And eventually, as the usage goes to zero, it diverges. There's a very simple argument that essentially it tells you that if the usage is sparse, the complexes are very much more orthogonal to each other, right? And there's a non-trivial exponent there because a little bit of tangram you can play, so that's why it's 3/2 and not 1. But you're gonna play too much, and that's why in the end you ought to do some of this sparse usage. Now, we don't need to do any database comparison to be sure about the sparsity of the proteome usage. We can just do a very trivial back of the envelope calculation. I mean, I kind of gave it for granted at the beginning of the seminar. So we've defined usages as our variable, right? So we have to estimate it as the number of components used by a typical complex over the proteome size. And we know the number of components used by a typical complex is smaller of the number of components of the largest complex, which is a ribosome, which has about 1/10 of the proteome size. And so the usage is at most being very generous, 0.1. And that tells us we're definitely in this sort of diverging region, right? So in some sense, what I'm trying to say is that we can hope or begin to give explanations for the properties of protein complexes, right? For this strange phase of matter that exists within the cell, and that I hope it is clear that this is not only interesting, I think, from the point of view of biology, which I think it is, right? It's certainly just a fundamental biological question, but also from the point of view of material science, if you wish, right? So protein complexes are different, right? Biological systems are unlike physics, right? to borrow what Freeman said, in several ways. So often it's emphasized that biological systems are small, that cells are small, and it's true that they're small, but there are smaller systems out there, right? And the beautiful physics experiments being done for these smaller systems. So yes, there's not an ice cube, but they're also not 12 atoms, right? So I think that's not what's more distinctive. What's more distinctive is that the number of different components that they have is very large. Okay, so that is very unlike physics, right? That you have as many different components as the size of the system, right? I mean that is really odd from the point of view of physics, and I think it has to do with the type of interactions that one generates in the end, and in that sense they borrow a little bit from glasses, right? Because it's true that glasses such as this one do not have many different types of atoms, but they do have many different types of interactions, right? But even then, I don't think it's the same, right? Because glassy systems are disordered and they're irreproducible, right? So they do have many interactions but they're irreproducible. Whereas protein complexes, they're highly reproducible, right? So you can assemble them, self-assemble them from their components in any context, right? So I think they lie somewhere in between. They have some properties of order materials, they have some properties of disorder materials. And they're more reminiscent of engineered systems such as DNA origami. So maybe I'll just summarize this and tell you 2 words of one possible future project, so you have some idea of some of the directions I'm thinking of. Yeah, you know, I guess I just want to say that protein complexes can be thought to some extent as a new regime of matter that is characterized by self-assembly, is bounded by another special regime of matter, which is these chimeras. They have heterogeneous composition, they have sparse proteome usage, they show weak combinatorial expansion. If you want to learn more about this, it's in the paper that Stan and I published at the beginning of this year. It was just some more, you know, a lot of simulations and data analysis and things like this that I didn't talk about today. In a way, what I think we were trying to do is to replace these complicated high resolution molecular structures of protein complexes by something very simple. By doing so, of course, you won't capture very accurately the properties of any individual complex, but we're hoping at least you will capture the structure of the phase diagram, right? And maybe even the properties of the phase boundaries. So now I'll just have 2 more slides, and I want to tell you a little bit of one possible physics, pure physics idea that one can go develop from here, right? And this idea of developing something similar to these protein complexes, these programmable solids, if you wish, but in the liquid state, right? So imagine that now we want to have liquids that have this type of programmable phases. So we know what's the maximum number of phases that we can assemble with a certain number of components, right? This is Gibbs phase rule, and it's very large. However, this has no constraints, and I'm more interested in the case in which we have a liquid with many components and taut, right? And we want to assemble phases that are constrained in how many components they have. Okay, so imagine they have NC components. The question now is how many such phases can you encode? Okay, this is a very non-trivial question. In a way, what we're asking is, can we play combinatorics with liquids reusing the components? Right? There's actually very little known about liquids with many components. I think there's only 3 people that have done something in this direction. You know, somebody in Madrid, Cuesta, then Dan Franco, a little bit, and a little bit a new PI and a new assistant professor in Princeton. But anyways, to this type of question, there's 2 answers, right? There's of course a trivial answer. It's just some linear problem. We cannot play combinatorics, so you just divide total number of components into components in a phase, and that's the number of phases you will have. Or maybe there's something non-trivial, right? And we can actually play combinatorics. So the starting point would be the mid-field free energy of a liquid and with many components. And of course, this is somehow reminiscent to the neural network models, right? And then you can try to program, right, in the interaction term, these phases. It's an open question whether you can actually play combinatorics. I used to think so, now I'm not so sure. I started doing some calculations. I think you may need something else than this simple free energy, right? But what we're aiming for is having something like liquid tangrams, right? So these programmable phases, and then other even more exotic regimes such as liquid chimeras, right? So this would be a multi-component liquid that has local. So it's like a compositional liquid crystal, if you wish, right? It would have local compositional order, okay, so local properties reminiscent of what we encoded, but overall disordered, right? So these are, I think, completely new phases of matter, really, and I think they're theoretically accessible. And I mean, I haven't thought too much about whether these are also one can engineer them, but this is definitely something to think about. So that's all I wanted to tell you about. Yeah, I mean, also we're, if there are students in the audience, I cannot see who's in the audience. You should know that we meaning our group, which is composed of just me at the moment, we are hiring, right? So there's possibilities for postdoc positions and also for PhD positions to start next year. We're located in this institute that you may or may not know here in Lisbon, and we have funding for traveling and to invite speakers. And in this context, I would also like to strengthen interactions with all of you. I hope you're still there. This is all I have to say. Thank you.
Okay, thanks very much, Pablo. And we now invite questions from the audience. So if you want to ask a question, just raise the little blue hand. If you don't know how to raise the little blue hand, just shout. So who wants to be first? Come on, don't be shy. Questions, comments, please.
So maybe I can ask a question.
Yeah, go ahead.
So maybe I was a little bit lost, but I mean, I agree with you what you said at the beginning, that okay, this is biology, not physics, but still biological system must obey physical rules like everything else. So proteins are formed by atoms, and atoms are formed by electrons, and so on. So in which part of your model is there some kind of connection to these more fundamental physics? Let's say.
Well,
it's just the binding energy. It's the only parameter where you make a connection to real physics, to known physics.
I don't know what real physics means, but
physics that we know. Okay.
I think what you're hinting at is, okay, so I'm not putting any molecular details of the composition.
Yeah, and then I'm not complaining about it. It's just you have to. So at some point, your model has to have some parameters that could be related, at least in principle, to those more fundamental things. So it's just the binding energy. So you encode every interaction, complex interaction between proteins and between in that binding parameter.
Yeah. Okay. So there's two parts to the question. The answer to the second is that do you encode everything in the binding energy? Is yes. We've encoded all the self-assembly properties in the binding energy, and we've encoded all the properties of complexes in the statistical ensemble to which they belong. To the first question, what are the connections to, if you wish, real properties of protein complexes? How many complexes there are in a cell, that is a real property, right? So the fact that it's a statistical property, right? It's an ensemble property. It doesn't make it, I mean, less real in the sense that
Yeah.
Right. So and the other, and maybe to go a bit further, I think in the line that you're pushing is maybe it's a bit naive to just put one. I mean, no, not maybe. It's obviously very naive to just have one parameter E, right, to encode interaction energy. So we know interaction energies are obviously variable, right? We've explored in a naive way, just for completeness, this variability in our paper, right? So we've considered an ensemble of binding energies, but I think there's more interest. A bit, one can push this even further because these binding energies, they emerge from microscopic interactions between interfaces of proteins, and so you could, and that also is a constraint, right? Because interfaces of proteins are not, we're in this case considering them in a way infinitely dimensional, but in reality they're not infinitely dimensional. Maybe they have 5 or 6 amino acids that interact with 5 or 6 amino acids, right? And there's only 20 such amino acids. So there's a finite alphabet of possible interactions that you can construct, and that does pose a constraint as well. And I think that's a very beautiful idea. Unfortunately, somebody has already given, I think, significant steps in this direction. So not for self-assemblies, but just for pair protein interactions. It's, so there was a little bit done by Eugene Shaknovich and perhaps more by Gerhard Hammer, who's in Germany, right, in Frankfurt. So yes, there are additional constraints arising from the microscopic origin of these interactions, if you wish.
Thank you.
Yeah.
Okay, maybe I can, I think I'm unmuted, yeah.
Yeah, go ahead.
Okay, so somewhere, somehow, the size of the complex, whatever, comes to play, because I mean, you showed the example where you have the full glass or you have the atomic. So how is the total size or the constraint between the sizes and the number of components? That's got to play a role. I mean, you don't find complexes in every structures.
Right. So so in our model, if you wish, it does play a role. I just didn't talk about it, but yeah. So there's this K max, if you wish. It's so you have three. You can redefine variables in different ways, right? So define heterogeneity, usage, and proteome size. You could have heterogeneity, usage, and size of each complex, because then you can obtain the proteome size by multiplying the others, right? So it's not an independent variable. So there is a scaling with the size, right? And there is a minimal size beyond which these complexes are not really stable, right?
Uh-huh.
And the larger they are, the closer you are to a thermodynamic phase, if you wish, right? And the better things will work. What happens is, so in a sense, these complexes are not, they're finite size relative to the lattice, size of the lattice, right? They're small. And that means that they have an evaporation time, right? So they're somehow stabilized by the surface, because the surface has less interactions than the bulk. But they will have an evaporation time, right? So eventually they can vanish or nucleate, right? So yes, size is important. I didn't talk about it. It's, we don't size scaling and all this in our manuscript. We didn't do like orders of magnitude. We just did like a bit over 1 order of magnitude of size scaling, because we were not super interested in it, and we kind of.
Exactly, but I think the interesting question is how, I mean, you can find these structures which are not ordered, they're not completely disordered, they are they actually hallmarks of these proteins, so they're important, and they happen to, I mean, you see them in the proteins for a given size. In the models, how is that reflected in any way? I mean, if you, or it doesn't matter, the models will reproduce these types of assemblies with these phases which are not fully,
It's a very
fully
good
classy, fully disordered, or they're not fully ordered or whatever. But these must be for a certain size. I mean, unless.
Okay, so in the, let's say in the cell, right, you find protein complexes of many different sizes. Right?
Mm-hmm.
There's a distribution. So in principle, the problem is, this is one of these places where the data is a bit, but what we have done is we've explored, so in our model, let's say, when you start by constructing the ensemble of complexes, right?
Mm-hmm.
And then from that, you construct an interaction rule. So where you can put this information is in the ensemble of complexes. We have done some of that, but again, similarly to the variability of the energy, just a little bit, right? So just a bit for completeness. So we've studied the role of variability. Essentially, we've taken a Gaussian distribution of sizes of complexes.
Mm-hmm.
So you have a typical complex, and then a spectrum of smaller and larger, and then you can see how the phase diagram will change using the variance of this distribution as a variable, right? So this we have done. It's a distribution of sizes of complexes observed in cell Gaussian. No, what is it? Hard to tell, right? There's just not such a comprehensive data set, but if anything, it will be broadly distributed. But we didn't more than Gaussian invariable variance. So we didn't check, I don't know, like power law distributions in sizes, for example, right? But all these are things that can be done, right? Essentially, it's just playing with different different ensembles, right? And the question is, what properties of the ensemble you're interested in, right? And then does this naive interaction rule account for them? And the other thing to do with is a naive interaction rule, right? Can you modify this naive interaction rule into something a bit more complex that may accommodate for may somehow push these boundaries in different directions, right? And I think the answer to that is also yes, right? So there's no reason why the interaction matrix should be binary, right? 0, or, I mean, well, obviously there's no reason, but there's also no reason why that should be the best solution, right? That is a solution that works to some extent, but I'm pretty sure you can adjust this value of E for each interaction depending on the properties of that protein. So proteins that are more promiscuous have weaker interactions. Proteins that are more, or something like that, right? Proteins that are less promiscuous, they have stronger interactions, and that probably pushes the boundary further.
Okay, just one last comment, which is related to it. Presumably what you call the liquid would be the fully disordered glass in some other real world. Is that right? I mean, what do you mean by liquid?
Remember liquid, so I talk about liquids twice, one within the context of what I've done, and one within the context of what I want to do. You mean of what I've done or what I want to do? So what I did...
liquid is more like a glass, I mean, dynamically, or what?
Okay, so no, so in what I did, it's not like a glass. So the chimera phase is more like a glass, right?
Yeah.
But it's not exact, so it's more like, so this is very similar to, okay, very similar. This has a strong smell of a Hopfield model, if that says anything, right? So essentially, it's not a fully glassy state because it has
nucleus, yeah.
Different points, right? The liquid phase is a pure liquid in the sense that the dynamics are fast, okay? Components constantly rearrange. And when you increase the binding energy, there's a transition, from the liquid to the chimera glassy phase, because there's a kind of glass transition there too. So that's about what I did, right? Then of what I want to do, I'm just talking about purely liquid phases. So just taking field free energy of a liquid, right? In which space now there's no geometry of the structures to be assembled. And then you would have a liquid glass, if you wish, in which the local compositional order, right? Things will be constantly reshuffling, like in a liquid. But at scale, there will be compositional order to some extent, right? I mean, that's just my guess, okay? Here I'm guessing exotic phases, if you wish, but I think something like this. So essentially I'm talking about a liquid system with many minima, if you wish, right? A liquid phase with the possibility of having many minima, which I think is something that at least I haven't seen in the literature. Okay, and definitely not for. I can probably assert not for compositional case, right?
No, because you probably need too many components.
Many components,
and then it is a mess. I mean,
it may be a mess, but it doesn't need to be a mess. That's the beauty of it, right? So it may have a programmable phase, right? Like it happens with neural networks and like it happens with self-assembly. There is a messy phase, but there's also an order phase in which what you programmed is what you get. Experimentally, it's obviously a disaster, right? But
yeah, okay. Thank you.
Yeah.
Right. Very good. Are there any more questions, comments, praise, criticisms?
No, congratulations on the baby. I mean, we seem to be having a baby boom around here, and they all come in advance. I hope the next one doesn't, for reasons. Anyway, congratulations. Everything went well?
Everything went well, and it's been, it's easier than we thought. So I think he's better than our daughter. He's more chilled. So
what's his name?
Samuel. I think he works
Samuel. Okay. Okay. Congratulations. Congratulations to your wife too.
Thank you very much, Margarita.
Right. Are there any more questions or comments? If not, then we shall wrap up for today. thank you again, Pablo, for a very informative seminar. Thanks to all in the audience. And we'll be back next week when we shall resume an old CFTC tradition, which is the Nobel Prize seminar. So we'll have Carlos Herdeiro from Aveiro telling us about the 2020 Physics Nobel Prize. So see you then. Thanks to all. Bye-bye.
Bye-bye.
Okay.